Thank you very much. Now, finally, I can get to talk about actual science. You think I like to talk about that other stuff. I actually don't love doing it. I mean, some of it is kind of funny, but the whole story is kind of sad in the end. So, actually, no, this is much more interesting, I guess. So, yeah. So these are these wonderful gaseous mediators that we have been studying for a while, and here they are, all four of them, and in an hour you will know everything that is important that needs to be known about them. Okay, I try. But before I get to them, I have to tell you what is it that we are studying on them, and why is it that that's what we are studying and not something else. And after working in this field for a long time, I came to the conclusion that really everything is about energy, okay? And I am pretty convinced about this. And I don't mean this kind of stuff, and also I don't even mean this kind of stuff, even though this is actually true. What I mean is that basically all our cells are dependent on energy production and energy consumption, and basically energy is carried by ATP molecules. The phosphate groups are pushed on it, and that registers as energy, and when the ATP goes to different parts of the cell, ATP is utilized, and of course all the functions that we think are important are essentially dependent on ATP. Without ATP, cells die. Without ATP, cells cannot function. And so, considering all of that, to me it is always surprising that cellular bioenergetics as a topic is still not as widely studied or as fashionable as, let's say, signaling or many other topics. So I don't think there are departments of cellular bioenergetics even. So it's a kind of, everybody knows it's important, but I'm not sure if lots of people study it. And just as a reminder, of course, that in our cells, we can generate, mammalian cells, we can generate two types of mechanisms that can produce ATP. One of them is basically fermentation. The ancient mechanism goes back to bacteria, of course, or lower organisms even. And then the other mechanism is, of course, oxidative phosphorylation through mitochondria. So, this is basic biochemistry, but this will be important later. And so I'm just going through to the basics. And one thing that's important here that everybody knows is, of course, that the oxidative phosphorylation makes much more ATP per molecule of glucose than the glycolysis. What is less known, and this sometimes people forget, is that glycolysis is much faster. So if you don't mind that you are actually generating a lot of lactate as a byproduct, then actually from glycolysis you can actually make quite a lot of ATP. It's just that a lot of it is wasted and a lot of lactate is also generated. But you can still make quite a bit, actually. So, of course, we have mitochondria in our cells, and this is the inner membrane of the mitochondria where ATP is made, and this is, of course, the famous electron transport chain. So electrons are pushed, and it's basically like charging a battery. So the electrons are moving through the complexes, and the electrons are coming from electron donors into the system. And the reason this is happening is because this electron movement is basically pushing protons from one side to the other. So it's kind of like charging a battery, and you create a proton gradient between the two sides of the membrane, and then this proton gradient is going to be harvested by ATP synthase. And ATP synthase is a really interesting molecule, and this is supposed to play and rotate, but it's okay, you can imagine that the thing rotates, and it basically pushes extra phosphates onto ADP. So ADP takes one more phosphate, and that is the hardest to push it on because there's already two phosphates and the charges make it hard. And this third ATP is really the one that is carrying most of the energy, and then the ATP molecules go around the cells. So ATP synthase, it moves actually. Okay, so you can see how the things are pushed together, and then they leave the ATP synthase. And of course, this system is fueled by the Sandor-Krebs cycle. For Hungarians, it's a Sandor-Krebs cycle. For everybody else, it's just a Krebs cycle. And you are basically breaking down sugars into smaller molecules, and you are generating electron donors that then fuel this electron transport that I showed you. And if it's not sugars but fatty acids, the fatty acids can also be degraded, and then they fuel the same system. So this is why we are eating, because we need to fuel the electron transport, and we need to make ATP. And of course, this was a fundamental discovery of Szent-Györgyi and Krebs. And Szent-Györgyi also, at the time, brought this very controversial concept that actually proteins can be what he called semiconductors. So he was already thinking about electron transport in protein systems, and at the time this was ridiculed, but now we know that actually this is one of the key functions of enzymes. They are conducting and managing electrons. So Szent-Györgyi clearly had a lot to do with this, and so did this guy, Peter Mitchell. He was really the one that figured out, put the whole thing together and figured out why is actually the electrons moving, why is the battery charging, and what is actually the role of ATP synthase. And this is not how you would build a machine that makes ATP. The only reason this is like this is because of evolution. And this is one of the quotes that is important, that nothing in biology makes sense except if you look at it through the mirror or through the light of evolution. And this is one other quote which is also pretty good. Evolution is a tinkerer, not an engineer. In Hungarian it's even better. Evolúció nem tervez, hanem barkácol. It takes existing things and puts it together the way it can. And you would not necessarily design something this way, but because of evolution, this is what happens. And this is exactly what's happened with mitochondria, of course, because mitochondria are actually ancient bacteria that got into this primitive cell through basically endocytosis, endosymbiosis, and these ancient bacteria are our today's mitochondria. And this is where this gas is coming. Okay, so we will get to the gases. This is where the gas is coming, because these ancient bacteria lived in a gaseous environment where all these gases were around. And so these bacteria, today's mitochondria, are still maintaining all of those capacities to handle these gases, utilize them, respond to them, and so forth. So it all goes back to this. And even the ATP synthase might go back to the flagellar mechanism, because some of these ancient bacteria, today's bacteria, also have these rotating mechanisms, and there is quite a bit of homology between flagellar rotating systems and ATP. So there's a lot of evolutionary stuff here, but for our talk today, what is kind of important is that, I don't know why this pointer is not doing its job, but you can see, and this is a very complicated slide, but you can see that oxygen emerged relatively late over the years. So before oxygen happened, there was a lot of hydrogen sulfide around and no oxygen. And this endosymbiotic process actually happened back then. So that's just something to keep in mind for now. And then the other thing, of course, to keep in mind is that there is a reason why our blood and our extracellular fluids are 0.9% salt, because this is actually the salt content of the oceans. And there is actually quite a bit of evidence, or suggestions at least, that early life developed in these deep sea hydrothermal vents without oxygen, but in the presence of hydrogen sulfide. And there are still these forms of life existing down there in this deep sea hydrothermal vents, which are actually utilizing hydrogen sulfide and sulfur chemistry, and they are not, utilizing the normal systems that the other cells or the other organisms utilize. So there is a lot of stories going back in time, and one more thing to consider is that it's one thing to have a unicellular organism, and it's a whole other thing to have a multicellular organism. And one of the big differences is that unicellular organisms can freely diffuse, and because the gases can diffuse from the outside to the inside, and they can reach all parts of the cell, unicellular organisms don't need to have systems to move things around, like oxygen around or any other gases or any other molecules. These big guys, including humans, of course, they need systems to move oxygen around. And this is what brings us to the circulation, of course. So this is really the only function of circulation, to bring the oxygen to the mitochondria. Because what I forgot to tell you is, of course, that this oxygen is the final electron acceptor in the system. So the electrons are flowing, and eventually they have to find a resting place. And this resting place is oxygen, and then you make water. So basically, the only reason we have the circulation is to carry the oxygen to the terminal point in the electron transport chain. And the only reason for the electron transport chain is to make ATP. So everything is really about ATP still. And so in like 5 minutes, I think I covered why we eat and why we breathe and why we have circulation. That's pretty good, no? And that also tells you that really these are the only things we are studying. Bioenergetics, mitochondria, and maybe the circulation, okay? Everything else is not important. So this is what we study, try to. And now we go back to these gases. So nitric oxide. But first, you know, the general rules. Okay, so what is it that we call mammalian gasotransmitters? They are small gaseous molecules, of course. They are freely permeable, that's because they are gases. They are, in order to call it a gasotransmitter, it has to be produced by our cells, our tissues, the endogenous. So they have to be produced. They cannot come from the outside. They have to have regulatory functions, so it's not enough to be a toxic gas. And this is pharmacology, that the actions of the endogenous molecule has to be mimicked, recapitulated by giving the stuff from the outside. And based on these criteria, the top 4 will make it, and I will show you why. But for example, oxygen is not a gasotransmitter because it's not produced, at least not by mammalian cells. Ammonia is also not a gasotransmitter, as we know, because it is produced actually, but as far as we know, there is no regulatory roles. It doesn't seem to have any roles that we are aware of. So these are gases, and they have some roles, but they are not gasotransmitters. The top 4 are gasotransmitters. And one thing about gasotransmitters is, don't have to memorize this, one of the things about gasotransmitters is that they don't obey these type of rules. So, you know, when we teach biochemistry, then we teach these very specific pathways. One thing is acting on the other, and then that acts on the other, and there is all these pathways, and everything is very specific. Gases don't do that. Other thing, when we teach pharmacology, again, we teach about specificity, you know, receptor, ligand, specificity, selectivity, they bind, they act, very, very sort of structure. Again, gases don't care about these type of rules. And this is just an example, just to show you, for example, hydrogen sulfide is acting on all of these pathways. Maybe I will mention some of them later, but it's not important what these things are. What is important is that they act on many, 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 many things simultaneously. So it's a totally different way of thinking, and it's a totally different way of regulating. And so when I explained all of this to ChatGPT, I said, okay, make a cartoon. It made this cartoon, which I think is pretty cool. They all follow the central dogma. Okay, so let's start. So nitric oxide. First of all, I'm going through the order of discovery, let's say. It is a gas, definitely. It has all the characters of a gas. You know, it has a melting point, it has a boiling point, it can be detected, blah, blah, blah. And for a long time, it was thought that this is something that is only present in the environment. One of the ways you produce nitric oxide is actually through engines, you know, cars, and it's in the exhaust fume, and there is a higher nitric oxide level in the cities than outside the cities. And when you take your car for a service, this is one of the things that they measure, what is the emission of this stuff. And actually, the people who first tried to measure nitric oxide generation from mammalian cells actually used one of these machines. They took a machine that these car people were using to measure nitric oxide emissions, and they changed the sensitivity of the machine, and they reconfigured it for biological measurements. So this is actually how the first measurements were done. So this was the first nitric oxide, and they told you, it's a toxic substance, it's a gas, it's produced by cars, okay. And then the other thing that was clear also, that it's produced in the environment, that lightning, high energy lightning in the atmosphere can also produce nitric oxide. And actually this was an argument early on against the idea that our cells can actually produce nitric oxide, because the argument was, you need this huge energy, it happens with lightning, it will never happen in a cell. So that was actually an argument against it. They were all wrong, but that was what they argued. And this story actually started a long time ago with this very famous paper of Fribourg and Zavadski, which maybe now it's common knowledge, but they describe this really interesting finding that a blood vessel only relaxes to acetylcholine when the endothelium, the internal layer of the blood vessel, is present, and when you remove this internal layer, then you get no relaxation. And this substance initially was called EDRF, endothelium-derived relaxing factor. So it's interesting that in 1980, you could still publish a paper in Nature and say that you don't know something, okay? Now, forget it. So he called it EDRF, and for a long time this was like an open hunt for many people working in vascular biology. What is this EDRF? What could it possibly be? And this was, actually, believe it or not, this was the first scientific paper I ever read. I was doing research in a group in Semmelweis University, and this was the first paper that my supervisor gave me to read. And my English was not very good, so I had to go with translating line by line. And this is basically a concept that says that if you have the endothelium present, and then you have thrombocytes trying to aggregate, if the endothelium is there, the substances that are released from the thrombocytes are going to cause a relaxation, and the thrombus will be flushed away. But if the endothelium is not there, then these substances will directly contract the vascular smooth muscle, and this relaxing effect of the endothelial cell is not present anymore, and then you get spasm, vasospasm, stroke, heart attack, and so forth. And this concept is still valid. At the time, they still didn't know what this EDRF was that is mediating the relaxation. And then, bit by bit, it was more and more clear that there are many similarities between nitric oxide and EDRF, but still people in the field couldn't actually believe that it's the same thing. There was this time when they had all these articles that said, there are many similarities between the two of them. Yes, they are very similar. And then eventually somebody said, they are similar because they are the same thing. And that was, of course, the famous discovery of Fulvert, Ignaro, and Murad, and also Moncada was a part of it who didn't get the Nobel, discovering this pathway and the role of nitric oxide in it, and the mechanism through which this process happens, which is, of course, the cyclic GMP mechanism. So nitric oxide goes to the blood vessels, relaxes the blood vessel, and this is through activation of soluble guanylate cyclase. So this is not textbook stuff. And this concept of endothelial dysfunction is now also in medicine, and it underlies a lot of diseases, including hypertension and diabetic complications and many others. So that was me many years ago, and we were also already working on these endothelium-dependent responses back in the day, and this was one of my first papers ever. And this field actually evolved now into all kinds of directions. One of the important discoveries was that actually the enzyme that makes nitric oxide has 3 different isoforms: the endothelial isoform, the neuronal isoform, and the inducible or inflammatory isoform. And each of them have different regulatory functions in different systems. Then there was a huge advancement in the field showing that nitric oxide synthase can actually either produce nitric oxide in its coupled state, or it can also produce superoxide in an uncoupled state. So depending on how it is assembled and what are the cofactors, it can either produce something that is regulatory and beneficial, or it can produce something that is actually deleterious in most cases. And there was a lot of discoveries around the cofactors of these enzymes, the regulation of the enzymes, the electron transport. Remember, SENDRD here is an enzyme that is actually an electron transporter. And then there was a lot of discoveries around the brain, nitric oxide synthase playing a role in memory formation, and the inducible nitric oxide synthase playing a role in antimicrobial defense systems, anti-cancer responses, and other sort of inflammatory and immune responses. So by this time I was in the William Harvey, working with John Wayne, and at the time the famous advancement in the field was that there are the inducible forms of nitric oxide synthase and the constitutive. And the inducible form plays a role in different forms of inflammation, in circulatory shock. So we have been working on this, finding ways of modulating the system. And then we followed up on this later by demonstrating that the nitric oxide is actually not as toxic as you think, even at high concentrations, but it becomes really toxic when it forms peroxynitrite, which is this larger molecule, which is a combination of one molecule of nitric oxide and one molecule of superoxide. So we started to work on peroxynitrite, mediated responses later, and here is the formation of peroxynitrite. This is one of the many directions where nitric oxide can go. It can go into nitrite and nitrate, which are kind of semi-stable products, but nitrite and nitrate can actually go back to nitric oxide. In some cases, it can go to nitroxyl, nitrosyl, and others. But peroxynitrite seems to be one of the sort of downstream products of nitric oxide, which is present in inflammation, in reperfusion injury, and in different conditions where there is higher amounts of nitric oxide and also increased amounts of superoxide. So we did a lot of work on this field too, and through this peroxynitrite story, we ended up also working on this really interesting enzyme, polyadipyribose polymerase, which is another very interesting topic, which I'm not going to cover very much, but this is an enzyme that makes these polyadipyribose chains upon recognizing DNA damage. And basically, the original concept that was developed with contributions from our group and many others is that under certain conditions, when DNA strand breaks happen, PARP is activated and it actually builds these polyadipyribose chains in the nucleus, but at the same time it impairs mitochondrial function on the outside. And again, this kind of goes back to the energy story, because what's really happening here, and now we know this, at the time we didn't know, that the cell is prioritizing for DNA repair, and it pushes these polyadipyribose molecules into the nucleus because they are actually utilized as a substrate for certain DNA repair enzymes. We didn't know that at the time. Now it's clear. We had no idea why the cell is doing this, because PARP is not a DNA repair enzyme. But it looks like PARP is prioritizing ATP or ATP equivalents into the nucleus to have DNA repair, and at the same time, when there's too much of it, then on the periphery you have an inhibition of bioenergetics, and that is actually bad for you. So it can create cell dysfunction or cell death. So this is a whole other story. I'm not going to get into that in detail, but we have been working on this, and there is a lot of follow-up on this, and there are clinical drugs on this, and there are two world experts on PARP sitting right here in this room, and so you can talk to them about PARP any time you want. So let's go back to nitric oxide, okay? So nitric oxide is not just an interesting molecule from a biological standpoint, but it also is the foundation of several very important classes of drugs. And this is the timeline of how those drugs were discovered. One of them is, of course, nitroglycerin. Nitroglycerin, this is dynamite, and so this is explosive, but it's also a drug. And this is one of the ways you can deliver nitric oxide to your cells and tissues. Nitroglycerin is decomposed or metabolized through a variety of pathways. Eventually, it will give you free nitric oxide, so it will mimic the effect of nitric oxide. Nobel, okay, I'm not going to go through this. I'm going to go through this even though this is really interesting. What I want to show you is that the main reason Louis Ignarro was in the three people who got the Nobel is that his discovery helped with the development of Viagra. Okay, and there's this old joke that, of course, they gave the Nobel Prize to the guy who discovered the foundations of Viagra, because the Nobel Committee is mainly older men. And, but this is, again, it's nitric oxide, okay? Because what really Viagra is doing, it's not making more nitric oxide in your penis, but it makes the nitric oxide last longer, very simply. So it inhibits the degradation of the second messenger that nitric oxide is inducing. So it is very, very closely related to the nitric oxide pathway. And another application of nitric oxide that is in the clinic is the inhalation of nitric oxide. So it's a gas, you can get a gas tank, you can get a regulator, and you can make patients inhale nitric oxide. And what happens when you inhale nitric oxide? It goes to the alveoli, but it only goes to the alveoli that are open. So that's good, because that's where the oxygenation also happens. It passes through the alveoli, it goes to the blood vessel, it dilates the blood vessel. So it actually improves the blood flow of the oxygenated alveoli, so you get an improved oxygenation. Also, nitric oxide is a molecule that helps development of blood vessels, development of lung tissue, and one of the approved applications of nitric oxide inhalation is actually to give it to preemies. So early little babies that are born too early and they don't yet have a fully developed lung, and if you inhale nitric oxide if they inhale nitric oxide or you deliver nitric oxide, it improves the maturation of the lung, and this is one of the indications. The other is to improve oxygenation, for example, in critical illness. And this is actually important because there is not that many other ways to actually selectively improve oxygenation to our tissues. There's one other newer direction in this field of nitric oxide, and this is modulating downstream pathways. So downstream of nitric oxide, you have the guanidate cyclase, which I already showed you. Nitric oxide binds to it, and it activates it, and then you get cyclic GMP, and you get the response. But the guanidate cyclase can be oxidized in certain diseases, and it can even lose its heme, and the heme is the one that's binding the nitric oxide. And these oxidized and heme-free forms are no longer responsive to nitric oxide. But there is a way to make them active again, either by changing the oxidation form, or to actually replace the missing heme. And this is a new classes of drugs that either activate the oxidized form of the enzyme or completely restore the activity, because that one on the right actually folds together in real life, and it fits into the pocket which was missing the heme, and it kind of gives the guanidate cyclase a new heme, and then it's responsive again to nitric oxide. And so this is a new class of drugs, which are, some of them are approved, some of them are still in clinical trials, but this is a new direction in nitric oxide research. And one last thing about nitric oxide is that it regulates lots of functions in the cell through lots of different mechanisms, and one of them is the guanidate cyclase mechanism, but another one is nitrosylation. So you have on proteins, you have cysteines, which have these free SH groups, and these free SH groups can be modified by a variety of mechanisms. They can be oxidized, they can be hyperoxidized, nitric oxide can nitrosylate them, and when that happens, the function of the protein or the enzyme is altered. So this is what I showed you at the beginning, that these gases can react with many targets, and they can kind of reprogram our cells. So this is what nitric oxide does, and it turns out that there are actually enzymes that catalyze these reactions, that actually put the nitric oxide onto these SH groups, and there are also enzymes that are removing these NO groups from the proteins. So either they put them on or they remove them. And this is again a new line of thinking about modulating and reprogramming the cell for therapeutic applications. So one of the new ways of doing this is actually to inhibit the denitrosylase, and that way you are increasing the nitrosylation of different proteins, and that is typically a good thing in several cardiovascular and other diseases. So this is nitric oxide today, and I even forgot the substrate, so it's coming from L-arginine, of course. I discussed S-nitrosylation, I talked a little bit about mitochondrial effects, reactions with metal centers. Bear-shaped dose response always happens with these gasotransmitters. So at low concentrations, they do one kind of thing. At high concentration, they do a completely different kind of thing. At low concentration, it is typically regulatory. At high concentrations, it is typically toxic. And it goes back all the way to bacteria. So bacteria also have nitric oxide synthesis. Okay, carbon monoxide, I'm going to go very, very fast with this because it is the smallest of the fields and a lot of the findings are still kind of not so clear at this point. But again, it is produced from heme oxygenase. It is different from nitric oxide because it does not react with cysteines, but it has all these other properties in terms of bell-shaped effects, mitochondrial effects, and so forth. So carbon monoxide, same story again. It's coming from the exhaust, it's coming from furnaces that are badly managed, it's coming from volcanic emissions, it's coming from different industries, forest fires, and so forth. But in our cells, it's coming from heme oxygenase. So it's part of the heme degradation pathway. Heme goes to biliverdin, bilirubin, and so forth. And in one of these steps, one of the products is carbon monoxide. It has been known for a long time, just nobody knew what to do with this finding. Nobody knew why it's happening or if this carbon monoxide plays any kind of role. But now we know that it has many biological roles. A lot of it are similar to nitric oxide. Again, there are evidences for being a neuronal messenger in the brain. Again, there are evidences that it can modulate anti-inflammatory effects, it can provide cytoprotective effects. The mechanisms that are underlying it are not so clear. I listed the soluble guanylate cyclase, but it is thousands and thousands weaker than nitric oxide. I listed the MAP kinase pathway that is definitely one downstream mechanism, NF-kappa B might be. So many pathways have been implicated, but overall the story is much less clear than with nitric oxide. There's different isoforms, heme oxygenase 1, heme oxygenase 2, different tissue distributions, a little bit like with nitric oxide synthase. And people are trying to develop new therapies out of this. So there are carbon monoxide donors, there is carbon monoxide inhalation, so they are kind of following the path that the people working in nitric oxide have laid out. There are many early stage clinical trials with carbon monoxide inhalation. The big problem or issue with this is that as soon as you start to mess with carbon monoxide, you're then going to end up with carboxyhemoglobin in your blood, and when you have too much of that impairs oxygen delivery to the tissues, so you have a limit of how much carbon monoxide is possible to give without some kind of risk. And these are still sort of in the early stages, but these are some of the compounds that can release carbon monoxide, again, in early stages, but this is a field that is still going. Third is hydrogen sulfide, and this is something that we have been working on quite a bit. Again, there are several enzymes that make it substrate is cysteine or 3-mercaptopyruvate, which is coming from cysteine. Again, I will show you that it can induce self-hydration, so another reaction that happens with cysteine, and it has all these other properties, the same as the others, bell-shaped curves, mitochondrial effects, and so forth. And where is the hydrogen sulfide coming from in biology? I already explained it's coming from the deep sea vent, so that has the evolutionary side to it. It is coming from different industries. It's coming from swamps. In the swamps, the source is bacteria, so some bacteria produce hydrogen sulfide. These are these septic tanks, again bacteria producing hydrogen sulfide. But in our body, all these enzymes are producing hydrogen sulfide. Now, what is hydrogen sulfide doing? Again, it's a bell-shaped story. So it has one kind of effect at high concentration, and one kind at low. At high concentration, it inhibits complex 4. So again, this is the inner membrane of the mitochondria. Electrons are running, and hydrogen sulfide can inhibit at complex 4 in a reversible manner. And when you have that, the electrons are no longer able to come, the pump is no longer working, and ATP is no longer produced. So you get basically an inhibition of the ability of your mitochondria to make ATP. It's reversible. So for example, if somebody falls into a septic tank and gets hydrogen sulfide poisoning, if they get it out of there, and many times what happens is that the people who go under to try to get it also get the hydrogen sulfide poisoning, so it's complicated. But if they somehow get all the people out of there and they end up in an ICU on life support, then just with supporting therapy, people can recover quite amazingly fast, because the hydrogen sulfide can diffuse off, and this system can be pretty well reconstituted. So this is the toxic effect. and in Japan, people figured out a way to mix different household reagents and as a convenient method of suicide, for example. Okay, I'm going to skip all of this. No, it's actually interesting. No, okay. These are the enzymes that produce it. And again, there is the old concept which says that one of these enzymes is more in the vasculature, another one is more in the brain and the liver, and the third one is everywhere, but it's mainly mitochondrial. And now we know that this concept is not really like this, because all of the enzymes can be in all of our tissues, and this last one is not just in the mitochondria, it's also in other parts of the cell, in the cytosol. But this was kind of the early concept, and it's kind of easy to remember, but it's wrong. But what's interesting is that in '96, Abe and Kimura published this paper saying that hydrogen sulfide is produced in the brain, and it has neuromodulatory and neuroprotective effects. And this was one of the early discoveries that started this field. This is Kimura, that is in Nauru, and behind us that's actually a volcanic place near Naples where the sulfur emissions are happening. And where did Kimura's idea come from? Kimura's idea came from toxicology studies. All these people that I mentioned that get hydrogen sulfide intoxication, and they measured hydrogen sulfide and thiosulfate levels in their blood. And they had, of course, in these studies a control group, healthy control as a baseline. And what Kimura noticed is that the healthy control is not zero, that there is hydrogen sulfide already in the healthy control group. And he said, okay, well then it has to be produced somewhere, it has to have a source. And since he was a neuroscientist, he was mainly interested in the brain, so he started to study brain, and that's how he came up with this idea. And this field really grew over the years. So now we just published a paper in our review in the physiological reviews. This is what you do when you are locked into your room during COVID. You write a review which is like 200 pages and 300. And this has over 2,000 references. So this is how the field grew, 2,000 references just on the physiology. That's not even the pathophysiology or the experimental therapy. And it's everywhere. It's regulating all kinds of functions in the body, from cardiovascular to neurological to genitourinary, brain, and so forth. So it's really a global regulator, kind of like nitric oxide. And it is regulated by production on the left side and also by degradation on the right side. And there are many different enzymes that make it, so the field has really grown into characterizing all these different enzymes, a lot of different reactions. And this is a brand new paper just coming out from our group, showing that actually it has a supramolecular organization, one of these enzymes, CBS. So it's actually a very complicated structure, so it has like stored pools of the enzyme that can be released and can be activated or deactivated depending on the need for it. So it got really complicated over the years. 3-MST also got really complicated, but the function is what I think is really interesting. So instead of this kind of function, inhibition, lower concentrations of hydrogen sulfide are actually donating electrons. So they are able to do what the regular electron donors like FADH, NADH can do. They can donate electrons into the electron transport chain, and they can actually support or at least compensate for electron transport and ATP generation. We cannot live on hydrogen sulfide alone, but if, for example, the glucose-derived electron donors are decreased, then the hydrogen sulfide electron donors can compensate for it, and you can have a sort of improved bioenergetics of a cell. So this, we think, is the physiological function of hydrogen sulfide, to stimulate or compensate electron transport, at least one of the functions. But there are actually several different functions. It can also open KATP channels through protein self-hydration. So what nitric oxide is doing, it nitrosylates them, so you get S and O. In this case, it self-hydrates or persulfidates them, so you end up with SSH. So again, it's a different kind of modulation, a different kind of modification, and it can affect thousands and thousands of proteins, and each of them can have a different function. So when you have a self-hydration of thousands of proteins, the cell is reprogrammed, signaling is affected, bioenergetics is affected, all kinds of functions can be affected. So it's a pluripotent regulator. It is just a partial list of things that can be self-hydrated, and it has things like KEAP1, which is part of the NRF2 pathway. So immediately this will explain why, for example, you have cytoprotective effects, antioxidant responses, preconditioning type responses. But there's a whole bunch of other things listed, and there's as I said, there are thousands more, and even the function of most of them are not known. Like, what is the effect of these modifications? We know that they are modified, but we don't know what they actually do. So you have SNO, you have SSH, and it gets really complicated from a really simple system. For example, when we just give hydrogen sulfide into a dish which has cells in it, you get secondary and tertiary species produced. So just from giving hydrogen sulfide to a cell, you end up with a mixture of all of these different species. And depending on what kind of cell it is and how you maintain your cells, these mixtures can create a variety of biological responses, totally dependent on the condition. So the hydrogen sulfide field got the first boost when there was this paper in Science suggesting that hydrogen sulfide can cause hibernation. And what they did was they put the mice into a hydrogen sulfide containing chamber, and they cooled the chamber, and the metabolism of the mouse decreased, and you could recover the mouse later. And so this was the big story, and it was picked up by all the popular media and everybody, until soon, and then later we and everybody else realized that you can do this trick with mice, you can do a little bit of this trick with rats, and you cannot really do it with anything bigger than that. So this kind of thing is only for very small animals and definitely not for humans. So this is never going to be translated as far as I can tell. But what can be theoretically done, one can do potentially hydrogen sulfide mediated suppression of metabolism, for example, for transportable organs, to store them, to be able to take them to another place, to have the longer, basically, half-life. So you can modulate metabolism this way. And this is being translated into clinical. And so we worked on this for quite a bit, and we worked out methods and donors and inhibitors and biochemistry and different pathophysiological and physiological conditions. And I'm not going to show all of them, but I will show a few of them really, really quickly. So one of the things that we worked on was diabetic complications. And it turns out that in diabetic complications, hydrogen sulfide levels are decreasing. It turns out that in diabetic blood vessels, you have an imbalance of reactive oxygen species and hydrogen sulfide and also nitric oxide, and the end result is a lower level of hydrogen sulfide and a loss of vascular protection, basically. So the same story what I showed you earlier, a loss of endothelial function. And so one of the offshoots of this is that one could replace hydrogen sulfide in diabetic blood vessels or in diabetic heart and improve relaxation or contraction of the heart in diabetes. And this is also being translated, so hydrogen sulfide donation is now becoming a potential therapy for diabetic complications. Another thing that is happening is that hydrogen sulfide is linked to cancer. And this is our work inspired by some reports early on that docs can sniff out cancer patients for some reason, which we think is now a signature which includes hydrogen sulfide from cancer patients. And we had this paper published more than 10 years ago now, showing that cancer cells upregulate hydrogen sulfide production, and they use hydrogen sulfide for a variety of roles. So cancer cells upregulate hydrogen sulfide production, and they use it for energetics, they use it for migration, invasion, and proliferation, and they also use it for angiogenesis. And the offshoot of this is that if you inhibit hydrogen sulfide production in a cancer cell, you can deprive the cancer cell from all of these benefits, and that can have anti-cancer effects. So this is something that we are actively working on, and I'm going to just skip all the slides here because of time, but there are clinical correlations, so high versus low CBS expressors have a different survival chances, and there are many, many mechanisms that hydrogen sulfide regulates in cancer cells, including stemness, including angiogenesis, including responses to immune therapy or to chemotherapy. And by inhibiting all of these, one can sensitize cancer cells or one can kill cancer cells. And this is just some in vivo experiments, those responses with different inhibitors. This is another in vivo experiment. You can actually cause tumor regression by inhibiting this pathway. And this is a sort of translational experiment. So depending on if you use patient-derived xenografts, if you transplant something that has high hydrogen sulfide generation, they grow faster and they respond better to inhibitor. If it's a cancer that has low hydrogen sulfide, they grow slower and they don't respond to inhibitor. So this is kind of being developed. And last but not least, we are working on Down syndrome, totally different story. But in Down syndrome, the triplication of chromosome 21 gives you an extra CBS. So in the brain and other tissues of people with Down syndrome, you have an excess hydrogen sulfide, which is actually deleterious for them. So this is basically the story. This came from original observations from Lejeune, who discovered it. Then Pierre Camun, who came up with this idea back in the days, but it was not really tested for a long time. And in this paper, we have actually used seahorse analysis to test this hypothesis in Down syndrome cells. Down syndrome cells produce more hydrogen sulfide. They are suppressed metabolically. One can restore the metabolism of these cells by inhibiting this pathway. And we did all the omics analysis and all the fluxomics analysis and all the biochemistry behind it. And basically what it comes down to is that in Down syndrome, the mitochondria are not functioning and the Krebs cycle is also inhibited, or it's even going in reverse. And so the cells with Down syndrome switch over from oxidative phosphorylation to glycolysis, but with inhibition of this pathway, we can restore it. And surprisingly, so a normal cell, mainly oxidative phosphorylation, Down syndrome, mainly glycolytic, and with inhibiting hydrogen sulfide, restoration can happen. And this can actually be translated into in vivo experiments when Down syndrome mice or Down syndrome rats are treated with these inhibitors and they are improving in their brain function. So this is a project which is going into clinical trials next year, hopefully. And then, so this is kind of the concept. So you have a normal cell that has some level of hydrogen sulfide, but you could say the same for nitric oxide. You have some conditions like diabetic complications when there is less hydrogen sulfide. So in these conditions, the way to go is donating hydrogen sulfide. Then there is the cancer cell, which optimizes its biochemistry to make as much hydrogen sulfide as is good for them, not for us. And so the thing to do here is to inhibit it and kind of get rid of these beneficial factors for the cancer cell. And then in Down syndrome, you have even more hydrogen sulfide in the wrong places, and that suppresses the metabolic function, and then you can inhibit that and restore function again. So this is kind of the story with hydrogen sulfide. And for the last 10 minutes, I'm going to show you a little bit on hydrogen cyanide, okay? This is brand new stuff. And it turns out that hydrogen cyanide is also produced. Cyanide is also produced by our cells and tissues. And of course, cyanide has the worst history of all of these gases. Nitric oxide is pretty bad, hydrogen sulfide is pretty bad, cyanide is the worst. World War II, Agatha Christie, all the horrible stories. But, and of course, cyanide is an inhibitor of complex 4. This is why it's so toxic. And it's not reversible. So the hydrogen sulfide effect is reversible, cyanide is irreversible. And but if you look at the broader picture, it turns out that there are some plants that actually produce cyanide, and there are bacteria that are also producing cyanide, and they use it for quorum sensing and other functions. And so, same idea what Kimura had, we also looked at toxicology reports, and guess what? We all have a basal level of cyanide in our blood. So the toxicology reports, when you look at the control group, show about half a micromolar, 500 nanomolar cyanide level basally, and cyanated proteins even. So because of this, we started to look into, and because of some evolutionary considerations, first we started to look at whether cyanide can also have a bell-shaped curve. like all these other gases, because people always look at the sort of the high concentrations of cyanide. And it turns out that if you go down to sub-micromolar concentrations, then cyanide is no longer toxic, but it can actually stimulate mitochondrial function. So that was the first clue. From the outside, we give cyanide, and it can actually have some positive effects. But it was still not cyanide produced by us. So the real question was, is it possible that cyanide is also produced by our cells and tissues? And this paper came out last year. It's only Nature Metabolism because one referee in Nature couldn't be convinced that there is no way to differentiate between the different dissociation forms of cyanide. They didn't know basic biochemistry, and that killed Nature, and then we put it in Nature Metabolism. Anyway, so this was the paper that shows that cyanide is actually produced by mammalian cells, and it plays regulatory roles. And because I always give the other talk about replication and reproducibility, so here, for example, I had to kind of follow this advice that this is extraordinary claim, so let's try to prove it by extraordinary evidence. So we developed several different methods to try to measure this cyanide. We collaborated with leaders in the field of cyanide toxicology. These guys, Gary Boss and Brian Lowe, they have been working on cyanide for ages in the context of toxicology and antidotes and measurements. And so we got to talk to them, and we got to convince them that we are not completely crazy. And so they gave us some methods, we developed some other methods. This is one method. This is the simplest one. It's basically an electrochemical method. You are measuring cyanide using an electrode, okay? You need to alkalize your medium because otherwise the cyanide diffuses out, and then you stick in the electrode and measure. You calibrate it, and it's a selective electrode. So this was one way to do it. Another way to do it was Brian Lowe's method, which is a derivatization method and then mass spec. So in this method, NDA and taurine reacts with cyanide. You end up with this product CBI that you measure with mass spec. So this is the clinical use. This is what people use for toxicology studies. So we use this too. And then the third method, which came from Gary Boss, was this method, which is this two-chamber method. So you have these two chambers. In one, you put your sample, and the other one, you put a solution of this kind of scavenger. And when the scavenger reacts with cyanide, there's a colorimetric change. And between the two chambers, there's a hole. So if cyanide is produced, then the cyanide goes into the gas phase, it goes into the other chamber, it's closed from the top, and it bubbles into the other solution, and it creates a color change. So with all of these methods, and we also did confocal and some others, with all of these methods, we were able to show that cyanide is actually produced by mammalian cells and tissues. And we also measured levels in the blood, and here are the levels, so it's detectable. And this is in terms of nanomole per milligram tissue, but it converts to about 300 nanomolar in the blood, and it's in the whole blood, and most of it is actually bound to hemoglobin in the form of cyanomethemoglobin. And these are some tissue homogenates, and all of them produce cyanide, but we realized very, very early that if you homogenize them too well, then the cyanide production will decrease. And this was already one clue. Then we also did some precipitation experiments, and they showed that this is coming from some kind of protein, so it's enzymatic. And of course, we then tried different amino acids. And the only amino acid that stimulated cyanide production was glycine. So when we took our homogenates and we added more glycine, the cyanide production increased. And so different cell lines were tested, and they all produced variable amounts, but all detectable amounts of cyanide. And so it's coming from glycine. And the mechanism is really a reaction of hypochlorite with glycine, and hypochlorite is produced in all our cells in small amounts. And this is really the mechanism that we have demonstrated. And actually, this mechanism is known. So if you buy glycine already in the NSDS sheet, which nobody reads, it says don't mix it with chlorine bleach because you might end up with toxic gases. Toxic gases means cyanide. So this is a known chemistry, but nobody knew that this can actually happen in cells. So we did some confocal imaging with a cyanide dye that picks up cyanide, and we started to see these dots, that there is these dotted areas in the cell where it seems like there is more signal. And these dotted areas co-localize with lysotracker. So it turns out that cyanide is actually produced in the lysosomes for the most part, and then it diffuses out. So then we learned how to isolate lysosomes, we measured cyanide production from the lysosomes, and basically this is the mechanism through which cyanide is produced in mammalian cells. So it's produced in the lysosomes, it's produced through the reaction of bleach with glycine, and the bleach or hypochlorous acid is produced from peroxidases, and the two peroxidases that are most prevalent in the lysosomes are myeloperoxidase, depending on the cell or tissue, and peroxidazine. So you need the peroxidases, you need the glycine, and you need the acidic pH of the lysosome. So this is the reason why nowhere else is this happening, because these reactions are acidic dependent. And so then we figured out all these steps, and also we figured out that then the cyanide is decomposed by an enzyme called TST or rhodanase, which is a known enzyme. It's just that nobody knew why do we have an enzyme that decomposes cyanide if you don't actually produce cyanide. It turns out we produce it, and we also degrade it. And so this is really exciting to us because it's one more post-translational modification. Again, hundreds or thousands of proteins become cyanylated by cyanide, and the list is long, and all of these proteins seem to have very important roles in different metabolic or signaling function. So this is a work in progress, trying to understand what is the role of cyanide. One thing we have seen very, very clearly is that it stimulates bioenergetics. Again, it drives the Krebs cycle, it drives electron transport, it drives ATP generation, and it also shifts the metabolism towards lipid oxidation, so beta oxidation of fatty acids. And so we're going back to the secondary Krebs cycle, so it shifts to the left side. So it's more fatty acid derived acetylcoenzyme A, and then feeds into the electron transport chain. And so we are working on this. We are also trying to understand if this cyanide pathway plays a role in blood vessels. Answer is, it looks like it does. It looks like endothelial cells are also producing it, and it has protective roles. I'm going to skip the original data on this, and we are almost done. And of course, we all looked at Down syndrome because we have the models, and it turns out that it's also produced in Down syndrome. So now we envision Down syndrome as this mixture of toxic gases in the brain of these poor individuals, cyanide and hydrogen sulfide at the same time. And that's not a good news when you think about complex 4, and the two gases are actually synergizing in inhibiting this pathway. So this is the last slide maybe. So we have these 4 gases, and all of them have essential regulatory roles in bioenergetics, and also in most of them in the regulation of blood vessel tone and blood flow and cardiovascular. And they have roles in health, and they have roles in disease, and in each case one can modulate them by either donating them or inhibiting them, and that can have different effects in different conditions. This is how many papers are published on the field. So nitric oxide is the big one still. It's decreasing, but still the most active field. Carbon monoxide is a big field, but most of it is toxicology. Hydrogen sulfide is a big field, but not as big as nitric oxide. Cyanide is almost not even showing up on this, so it's a very, very small but growing area. And I already explained these bear-shaped curves, which were, of course, discovered by a Swiss scientist named Paracelsus. And I'm not going to explain this, but basically we see the biochemistry of the cell in this way. So you have all the structures, all the enzymes, all the pathways. But on all of these structures, we have these switches, these SH groups. And these SH groups can be modulated by all of these gases. They can be attacked by nitric oxide, by cyanide, by hydrogen sulfide. And we are still trying to figure out what this means in terms of relative potency, what happens when you have simultaneously all of these gases, what happens to the function of each of these enzymes that we study in our model systems, and we study metabolic enzymes for the most part right now. So we are trying to figure this out, whatever time still is left for us. And these are our people whose head is full of these poisonous gases and trying to figure it out. And this is where we do it. It's a place called Fribourg, which is not Freiburg, which is in Germany, but this is in Switzerland. It's between Bern and Lausanne. And we have buildings. And we also have this type of ugly buildings, which is where the research is done. And we have money from different places to do this. And actually, we are organizing a meeting in July for hydrogen sulfide, but there's also going to be a cyanide session. And we have 150 people registered, so it's still a growing field and growing, as opposed to the last nitric oxide meeting that had to be cancelled because there was not enough interest and they switched online and it was hardly a meeting anymore. So this field is still active, and we think that the cyanide is going to be the next wave of activity in this field. And we are still excited about it. Again, book recommendations, definitely St. George book, Peter Mitchell, Life, very, very interesting. This is a really cool book about mitochondria. Read this if you only have money for one of them. And this is a very, very good book also about medicine in general. And that was the last slide. So thanks for listening. Thanks for having me. And I know this was very, very fast, but I'm very excited about all of this stuff, and I wanted to show you as much as possible. And please ask any question, okay? Thanks for having me.